Bismillahirrahmanirrahim. Dear students, we discuss biological functions of cellulose and cell wall polysaccharides. First of all, key concepts. Key concepts consist of cellulose, structure of cellulose, properties of cellulose, functions of cellulose, significance of cellulose. Then after this we discuss cell wall, structure of cell wall, layers of cell wall and functions of cell wall. What is cellulose? It is the most abundant naturally occurring organic substance. It is the main structural component of the cell wall. It is the reinforcing material in the wood. Cellulose representing about 1.5 multiplied by 10 raised to power 12 tons annual biomass production. It is first described in 1838 by French chemist CHEM2114. It occurs in almost pure form in cotton fiber at around 98%. It forms the primary structural component of plants and is not digestible by humans. Torrance in 1895 and Nestokoff in 1900 recognized that cellulose is a linear polymer of glucose by X-ray pattern analysis. In 1920, it is discovered that cellulose is in the lignified wood pulp, flax, rami and cotton. The Chinese discovered the usefulness of cellulose and developed a process in AD 100, means about 500 to 2000 years ago, that gave them their most wonderful of inventions such as paper, creates other products such as rayon and the transparent film called cellophane. In vascular plants, cellulose is synthesized at the plasma membrane by Rossett terminal complexes. Cellulose is an important structural component of the primary cell wall of green plants. Many forms of alga and oomycetes. Some species of bacteria secretes it to form biofilm. Cellulose is an example of a natural polymer. It is a long chain polymer of glucose molecules joined together. As the plant adds one glucose molecule to the polymer, one molecule of water is released. Cellulose is the most abundant form of living terrestrial biomass. Here are some definitions. Pyranose. Pyranose is any cyclic isomer that has 5 carbons and 1 oxygen in a ring of 6 atoms. Furanose, any cyclic isomer that has 4 carbons and 1 oxygen in a ring of 5 atoms. Cellulose. Cellulose consists of 2 glucose molecules linked by a beta-1,4 bond. In cellulose, a sugar with a pyranosic structure is present. Physical properties of cellulose. Cellulose is non-toxic, biodegradable solid and homobiopolymer. At pure state, white in color. Molecular mass is around 1.44 multiplied by 10 raised to power 6 to 1.8 multiplied by 10 raised to power 6 gram. Density of cellulose is 1.52 to 1.54 gram per square centimeter at 20 centigrade. Cellulose has high tensile and compressive strength. Cross bonding gives the maximum strength. Distance between the carbon-carbon bond and carbon-oxygen bond is 1.9 to 2.1 angstrom respectively. Thermal properties. Cellulose is very much stable in thermal conduction. Cellulose shows thermal softening at 231 to 253 centigrade. It is not a thermoplastic polymer. Electrical properties. Cellulose shows very low conductivity and high resistivity of electricity. Pure cellulose shows maximum resistivity and water content decreases resistivity. Relative humidity also affects the electrical resistivity. Chemical properties. Its degree of polymerization is 8,000 to 10,000 units. Isolable is water, but uptake water around 8 to 14% at 60% relative humidity and 20 centigrade. Soluble in organic chemicals. Give anhydrous reaction with concentrated acids. Reacts with hydrocarbons, alcohols, ketones, acids, ester, amides, halogenated hydrocarbons and hydrazines, etc. Reacts with strong base. Reacting order is lithium hydroxide is greater than sodium hydroxide, is greater than potassium hydroxide, is greater than rubidium hydroxide, is greater than cesium hydroxide. For cation, the order of reactivity is potassium ion is the lowest and zinc ion is the highest, whereas for anions the order is chlorate ion is the lowest and thiocyanate ion is the highest regarding order of reactivity. Now, structure of cellulose. It is a homopolymer of glucose. It is a carbohydrate composed of carbon is 49.39%, oxygen is 44.4% and hydrogen is 6.17%. The molecular formula is N molecules of C6H12O6, N branch chain polymer of glucose. Cellulose is a polysaccharides consisting of a linear chain of several hundred to many thousand beta-1,4 linked D-glucose units. Cellulose is thus a polysaccharide, Latin for many sugars. Several of these polysaccharide chains are arranged in parallel arrays to form cellulose microfibrils. The individual polysaccharide chains are bound together in the microfibrils by hydrogen bonds. The microfibrils in turn are bundled together to form macrofibrils. Cellulose microfibrils in plants appear to be 4 to 10 nanometer wide under the electron microscope. Here in this slide you can see how the cellulose is formed from beta-glucose monomer. The cell wall of a leaf plant, which is composed of cellulose microfibrils in a plant cell wall, and microfibrils in turn to form cellulose molecules, and cellulose molecules are arranged by beta-glucose monomers, as you see in this diagram. Now, what is alpha D-glucose? When the straight chain form of glucose closes to form a ring, it forms a new chiral center at carbon number 1. To assign the alpha or beta configuration to these forms, you look at the relative position of carbon number 6 and OH group on carbon number 1. In the alpha form, as you see the structure below, the hydroxyl group on carbon number 1 is on the opposite side of the ring from carbon number 6. Beta D-glucose. In the beta form, structure number 2 as shown below, the hydroxyl group on carbon number 1 is on the same side of the ring as carbon number 6, as you see in the structure. When alpha glucose molecules are joined chemically to form a polymer, starch is produced, and similarly, when beta glucose molecules are joined to form a polymer, cellulose is produced. Two isomers of aldohexose sugars are known as glucose, of which only one D-glucose is biologically active. The D-glucose can exist in two forms. One is alpha D-glucose and the second is the beta D-glucose, as is shown in the next slide. They differ only in the direction that H and OH groups points on carbon number 1. When beta glucose molecules are joined, a polymer cellulose is formed. Glucose, the formula is C6H12O6, is a monosaccharide or simple sugar. It is the most important carbohydrate. Cell use it as a source of energy and is a metabolic intermediate. In addition, glucose is one of the main products of photosynthesis and starts cellular respiration in both prokaryotes and eukaryotes. The mirror image of the D-glucose and L-glucose cannot be used by cells. In solutions, the open chain form of glucose, either D or L forms, exist in equilibrium with several cyclic isomers, each containing a ring of carbon closed by an oxygen atom. The D-glucose can exist in two forms. One is alpha D-glucose, which you are showing on the right side, and second is the beta D-glucose, which are showing on the left side in the slide. To assign the alpha or beta configurations to these forms, you look at the structure, look at the relative positions of carbon number 6 and OH groups on carbon number 1. In the alpha form, structure 1 as shown on the right side, the OH group of carbon number 1 is on the opposite side of the ring from carbon number 6. In the beta form, as shown on the left side of the structure, the OH group on carbon number 1 is on the same side of the ring as carbon number 6. In other words, at carbon number 1, if the OH group attached to it is below the ring, the molecule is alpha glucose, and if the OH group is above the ring on carbon number 1, the molecule is beta glucose. Here, the pyranosic structure of glucose. You look at the structure on the right side. At carbon number 1, if the OH group attached to it is below the ring, the molecule is alpha glucose. Now you look at the left side of the structure. If the OH group is above the ring, the molecule is beta D-glucose. Formation of cellulose from glucose. In cellulose, two molecules of glucose are joined to form 1,4-D-glycopyranosic bond, and a water molecule is released, as shown in this slide. Inter and intra-hydrogen bonding in cellulose. The red circle shows the intra-hydrogen bond, whereas green circle shows the inter-hydrogen bond in the cellulose. Intra-hydrogen bonding means bonding within a cellulose chain, whereas inter-hydrogen bonding is the hydrogen bonding which occurred between the two cellulose chains as shown in the slide. Crystalline and amorphous region. Here you see the structure in which hydrogen bonding within and between cellulose molecules. You can see crystalline region, intra and intermolecular bonding is so strong, whereas crystalline region do not absorb much moisture, whereas on the opposite side, amorphous region, bonding is weak and amorphous region absorb too much moisture. Cellulose formation in woody plants. Cellulose formation in woody plants consists of many steps which start from photosynthesis. Water molecules react with carbon dioxide in the presence of sunlight add chlorophyll to produce glucose plus 6 oxygen molecules. Photosynthesis is the first step, then sap conduction, then plasma membrane, rosette terminal complexes, spines, microfibers into cell wall, and the last one is the cell division of cellulose formation in woody plants. No reaction with water. Cellulose soak water very slowly. It uptake 8 to 14% moisture. Water is attached with strong hydrogen bonding. Relative humidity and temperature is important. And the last one is crystalline cellulose uptake less. Chemical properties. The hydroxyl groups of cellulose can be partially or fully reacted with various chemicals to provide derivatives with useful properties. They have the general formula cellulose OR, where the oxygen can be any of the cellulose hydroxyls. Cellulose esters and cellulose ethers are the most important commercial materials. Among the esters are cellulose acetate and triacetate, which are film and film forming materials. Functions of cellulose. Cellulose is mainly used to produce paperboard, cloth and paper. Small quantities are converted into a wide variety of derivatives produced such as cellophane and rayon. Conversion of cellulose energy crops into biofuels such as cellulosic ethanol is under investigation as an alternative fuel source. Cellulose for industrial use is mainly obtained from wood pulp and cotton. Some animals, particularly ruminants and termites, can digest cellulose with the help of symbiotic microorganisms that live in their guts, such as trichonympha. Cellulose has many uses such as the following: anti-cake agents, emulsifier, stabilizer, dispersing agent, thickener, gelling agent, and most important use is of the holding on to water. Significance of cellulose. Microcrystalline cellulose used as binder disintegrant in tables. Methyl cellulose used suspending agent and bulk laxative. Oxidized cellulose used as hemostat. Sodium carboxymethyl cellulose used as laxative. Cellulose acetate used as rayon or in photograph film or in plastics. Cellulose acetate phthalate used as nitrocellulose used as explosives, collodion or pyroxylin. Treatments of cellulose. Cellulose comprises nearly half the dry weight of most plant material. Carbon fibers containing at least 90% carbon obtained by the controlled pyrolysis of appropriate fibers. The term graphite fiber is used to describe fibers that have carbon in excess of 99%. Large varieties of fibers called precursors are used to produce carbon fibers of different morphologies and different specific characteristics. The most prevalent precursors are polyacrylonitrile cellulosic fibers, petroleum or coal tar pitch. Cell wall. The outermost structure of most plant cells is a dynamic and rigid layer called cell wall. The cell wall is found in plants, algae and fungi. Animals and protozoa do not have cell walls. In plants, cell wall is located outside the cell membrane and provides these cells with structural support, protection, and also acts as a filtering mechanism. It also acts as a pressure vessel, preventing overexpression when water enters the cell. The major components of cell wall are cellulose, pectins, hemicelluloses, proteins, and phenolics. The cell wall is a biphasic structure consisting of relatively rigid cellulosic microfibrillary embedded in gel-like non-cellulosic matrix. The microfibrillar phase consists of only cellulose beta-1,4-glucan, and the matrix is composed of non-cellulosic polysaccharides, include pectins, hemicellulose, proteins, and phenolics. The microfibrillar phase is readily visible in electron microscope and is crystalline. History of cell wall. A plant cell wall was first observed and named simply as wall by Robert Hooke in 1665. In 1804, Carl Dulphy and J.H.F. Link proved that cells have independent cell walls. A cell wall is a structural layer that surrounds some types of cells situated outside the cell membrane. It can be tough, flexible and rigid, which provides cell with both structural support and protection. Cell wall layers. Cell wall thickness is 0.1 micrometer to several micrometer. Layers: primary cell wall and secondary cell wall. Primary cell wall is thin, flexible and extensible. Primary cell walls contain cellulose, hemicellulose and pectins. It is permeable. Its size is 30 to 60 kilodaltons, whereas secondary cell wall is thick. Present inside primary cell wall contains lignin for strength and density of wood. Cellulose fibers contain cellulose is 35 to 50%. Xylem is 20 to 35% and lignin is 10 to 25%. Here you see the structure of cell wall. Third layer is the middle lamella. It is the outermost layer between adjacent plant cells. It contains pectins. Middle lamella gives stability, made up of calcium and magnesium pectates. Formation of cell wall. Again we discuss middle lamella. It is the first formed from cell plate during cytokinesis. Primary cell wall composed of cellulose fibrils produced at plasma membrane by cellulose synthase complex. Microfibrils held by hydrogen bonds. Secondary cell wall constructed between plasma membrane and primary wall. Plasmodesmata, interconnecting channels of cytoplasm that connects protoplast. Middle lamella is discussed in detail. Middle lamella is the first layer to be formed when a cell divides. It is an amorphous intercellular layer between primary walls of adjacent cells. It is cementing layer made up of calcium and magnesium pectinate or pectate, which joins or glues to neighboring plant cells. It is absent on the free surface of plant cells and in plasmodesmata region. Middle lamella resolves in ripe fruits, which results in softening. Middle lamella can be dissolved artificially by means of acid treatment. Primary cell wall is discussed in detail. The primary cell wall generally a thin, flexible and extensible layer formed while the cell is growing. It is the first deposition product of protoplasm outside plasma membrane and is present inner to middle lamella. Primary cell wall is delicate, flexible and thin, 0.1 to 3.0 micron meter, and capable of extension. Its thickness increases with the growth of the plant cell. It grows by further deposition of wall material into the existing primary wall. Secondary cell wall. It is often deposited on inner side of primary walls after growth of cell stalks. It is 4 to 10 micrometer thick, rigid, non-elastic, permeable and made up of cellulose and lignin deposits. Secondary wall deposition is not uniform. Such unthickened areas are called pits. Pits of two neighboring cells form pit pair. Pits can be of two types, simple or bordered. Pit chamber becomes flask shaped due to deposition in the form of border. During the development of pits, the secondary cell wall may overarch the pit cavity, forming a border, leaving an inner opening called pit aperture. Such pits with border are called border pits. Two opposite border pits are called border pit pair. Eukaryotic cell walls composed of polysaccharides, chitin and cellulose joined by beta-1,4 linkage. Examples are fungal cell walls, algae, water, molds, slim molds. Fungal cell wall consists of chitin and polysaccharides. Matrix of three components: chitin, glucans and proteins. Algal cell wall consists of cellulose or glycoproteins. Its components are mannans, xylanes, alginic acid, sulfonated polysaccharides. Water molds consist of cellulose 4 to 20% and glucans. Simple molds composed of cellulose. Prokaryotic cell walls. Bacterial cell wall is the major component is peptidoglycan. Gram-negative bacteria consist of thin cell wall, whereas gram-positive bacteria contain thick cell wall. Functions of cell wall. Cell wall gives cell rigidity and strength. It acts as a physical barrier to the plant. It prevents cell swelling and bursting as a result of osmotic pressure. It promotes cell-to-cell signaling through the cell walls. Thank you.